Welcome everyone again to another episode of the ideaXme show, the show that profiles the human beings behind the really big ideas that are shaping our world and inspiring future creators, and for all those that like really great stories. I'm Ira Pastor, your health, aging, and longevity ambassador along on this journey. So on today's show, we are going to delve into the area of ontogenesis, which is broadly defined as the development of a specific individual organism or anatomical or behavioral feature of that organism from its earliest stages to maturity. And this encompasses the disciplines of both embryogenesis and organogenesis as well. As many people may be aware, they follow the scientific literature and press. This week, there was a fascinating study published out of Ann Arbor, Michigan, at the University of Michigan, on a new method for making stem cell colonies that in many ways mimic early parts of human development. This really interesting and novel technique holds possibility to improve the ability to investigate important questions about both maternal and fetal health related to embryonic development, birth defects, even miscarriages. The technique developed imitates certain stages of embryonic development that occur shortly after implantation in the uterus, when the amniotic sac begins to form and when stem cells would ultimately go on to become the fetus and make their first steps towards organization and patterning in the body. One of the lead investigators in the study, who is joining me on the show today, is Professor Dr. Deborah Gumucio, who is Professor Emerita Cell and Developmental Biology at the University of Michigan Medical School. Dr. Gumucio's laboratory studies organogenesis with a focus on the molecular control of ontogeny, development patterning, and homeostasis of the gut, which is also defined as lumenogenesis, as well as inflammation and the molecular pathogenesis of various diseases such as familial Mediterranean fever. She directs the program in organogenesis for the Arthritis and Musculoskeletal Diseases Center and serves on the core advisory committee of the Cancer Center. She's also on the operating committee of the Medical Scientist Training Program. Dr. Gumucio founded the University of Michigan Center for Organogenesis back in 1995 and served as co-director for the past several years. And that center is a multidisciplinary center that serves to bring together clinical, basic, and applied scientists in the study of organogenesis and its relationship to human diseases. Dr. Gumucio has served on numerous committees and programs, including the Biomedical Research Council, the Biomedical Core Advisory Team, the Dean's Advisory Task Force, the Graduate Program Committee of the Program in Cell and Molecular Biology, a Program Committee for Construction of Basic Science, Medical School Executive Committee, and the Dean's Task Force for Research. She has organized numerous international symposia for the Center for Organogenesis, is a co-author of translational research in discovering gastroenterology, organogenesis to disease, and in national and international arenas, she has served as a member of the Eukaryotic Genetic Studies section of the National Science Foundation, ad hoc sections and site visit teams for the NIH, associate editor for the Journal of Molecular and Cellular Biology, a tremendous list of accomplishments. And in addition to all of this, Dr. Gumucio is also the director of a fascinating project which combines art and science at the university called the Bioartography Project, which is a really elegant project that's involved in collating these beautiful microscopic images from University of Michigan researchers all over the campus to display at the annual Ann Arbor Art Fair, where faculty and staff and trainees interact with the public to really explain the scientific and health significance of the work that's going on, providing the public a really great understanding of what's going on in the research labs for a really better appreciation of the world that surrounds us. All of that being said, Dr. Gumucio, thank you so much for taking time to come on the show today.
You're very welcome. Thank you for the invitation.
Absolutely. Absolutely. So, we typically start off by giving our guests the floor just to talk a little bit about themselves for a while, your background, where you grew up, how you got interested in science, how you developed this interest in developmental biology, and ultimately, your path towards these rather groundbreaking areas of ontogenesis.
Well, I have to say that if one describes my path, one would have to say winding. I've never been an A to Z person. I started out, I was born in Saginaw, Michigan, and was raised there. Mm-hmm. And it was my high school biology teacher, Mr. Fred Case, who was actually nationally known for his work in orchid breeding. And he absolutely inspired me in the field of biology. And that was really, once I took his class and became one of his assistants, it was just obvious that was the field I wanted. But my direction wasn't particularly straight, as mentioned. So From there, I went to the University of Michigan, and I was counseled in high school to be, if I was interested in science, what I should do is become a teacher, a nurse, or a medical technologist. And I thought of those. Medical technology sounded interesting, and so I got into that program. Was lucky enough to get a scholarship for it that paid part of my tuition. So I finished it, even though in the third year or so I hated it. So I decided to finish anyway, so I didn't have to pay my scholarship back as a loan. And then 5 years later, went back to school and decided to get a degree in epidemiology, which was, I had some experience in going through hospitals trying to find where disease outbreaks might happen in the nursery or the burn ward or whatever, culturing everything, trying to find the source of the disease and the name of the bug so that we could actually determine what antibiotic to give the patients.
Sure.
And that was fun, sleuthing work. So I got a degree in epidemiology. However, at the time, I also took my very first classes in cell biology. That completely put me finally on the path I wanted. However, I had to work for 5 more years before I had the money to go back and get a PhD, which I finally did. All this education at University of Michigan, which is rather unusual. And then did a postdoc at Michigan with Francis Collins, who was the director of NIH.
Sure.
I was his second postdoc, and by the time I left the lab, there were 26 of us. But he gave me tremendous autonomy and was extremely supportive of my career. And I was able to get a faculty position at Michigan. And from there on, opened my own lab and began my own kind of journey down the actual scientific paths.
Can we talk a little bit about your work in luminogenesis, which is a new term to me, but it's fascinating in its own respect, the development of the early gut. And I'm I've always been very interested in the entire area of embryology and early ontogenesis and all these dynamics that happen early on in life, not just because of sort of their relevance to understanding, as I said early on, sort of everything that happens at the very beginning of life, but also these connections that, whether they're in patterning or tissue remodeling or other things that happen later on in life, there are these really intricate connections. And so, if you can go into sort of how, or sort of what the area is about, sort of what is involved in sort of the early stages of studying this dynamic, and then how that, because I know you study inflammation and gastrointestinal disorders as well later on in life, how these areas connect, because it's really fascinating.
Yeah, so when I started the lab in 1991, I was extremely interested in the apical border, the luminal border of the gut. And for the reason that, first of all, the surface of the adult gut, the actual surface area, is roughly the size of half of a badminton court. So it's enormous, and it has to be that big because we have to have the surface area that we need to absorb nutrients. So if you don't have that big of surface area, it's actually, you reduce it more than 50% and it's incompatible with life. So it's really important to understand what contributes to that surface area. And there are 3 major things. One is the very length of the gut. The gut in an adult human is over 6 feet long. And about 40% of that length is attained before birth. So during embryo development, there is a tremendous emphasis in the embryo placed on trying to make this very, very long tube. In fact, it grows so rapidly that it actually spends part of its life outside of the body, per se, and then gets drawn back in to the body and bound in the proper way so that we can digest food and it doesn't kink up normally. So that's one thing, the length. A second thing is the fact that the entire surface of the gut is filled with these finger-like projections, and these are called villi. Each villus is kind of a structural, functional unit. which allows absorption of food. So you can imagine this finger sticking up there. The food is passing along here. This is lined by just one layer of epithelial cells.
Mm-hmm.
And in the core of it would be a blood vessel, lymphatics, a muscle to actually let it pump. And that whole unit basically is able to take in the nutrients from anywhere along the lining. So that, as you can see, amplifies the surface area.
Mm-hmm.
On top of that, if that weren't enough, each one of these individual villi has microvilli. And there are thousands of microvilli on every single cell. So these are relatively long projections of the cell itself.
Sure.
In tiny little, you might think of them as hair-like or bristle-like structures. So the whole organization, the structure is beautiful. I fell in love with it the first time I saw it. And so we studied multiple aspects of both lengthening and villus formation. And then at a time when we were trying to get into studying microvillus formation, we got off on another tangent that we'll probably talk about later, which is the formation of lumens in human embryonic stem cells. But for the intestinal, for the lengthening study, we've recently had some really interesting work that was done by Sha Wang, who's now at Duke University doing a second postdoc. Sha was a really energetic and brilliant investigator in the lab who discovered a novel, not exactly novel in the sense that it hadn't been described, but novel in the intestine of cells. So the early intestinal surface before you have villi is flat. The intestinal surface is flat. You have to make the villi during development. And in that flat intestine, it's still very actively growing. The cells are just multiplying like bananas. It's amazing. They just keep going. The cells are very tall. They're very thin, and their nuclei go up and down and up and down and up and down within the plane of the epithelium in a process called interkinetic nuclear migration. So they actually synthesize DNA when they're down at the basement membrane.
Okay.
Then they move up, and then up at the apical surface is where they undergo cell division.
I see.
And then they move back down to undergo another round. And it's very active. The cell cycle is like 16 to 20 hours, very, very rapid. So what Sha realized is that when the nucleus goes up and the cell divides, only one of the two daughters actually maintains a contact with the basement membrane. The other one is left pretty much untethered initially. And it has to find its way back down by sending a thin process all the way back down to the basement membrane. And what she discovered was the guidance cue for the podium to know where the basement membrane is. And that guidance cue turned out to be Wnt5a, which is a non-canonical form of Wnt. And without that, when she knocked Wnt5a out, the intestines were extremely short. They didn't grow because that daughter cell didn't know how to get back to the basement membrane and reconnect. It was lost by apoptosis. And not only is the daughter lost, all the potential daughters of that daughter would be lost.
Sure.
So it has a tremendous amplifying effect in a very rapidly growing epithelium to have that pathfinding go wrong. So that was really fun and interesting that was recently published. The other part of the story is after this lengthening process, during villus formation, there continues to be lengthening, but it isn't by the same way. It appears to be that, again, you're getting villi growing. The epithelium continues to be proliferative, and we haven't found exactly what is responsible for the lengthening other than continued proliferation. But what we have found is how the villi grow. And that turns out to be an equally cool story in that the epithelium secretes two ligands, both hedgehog family, Sonic hedgehog and Indian hedgehog, great names. The name hedgehog for this particular protein was actually first determined in Drosophila.
Okay.
Some of the early experiments showed that when you knocked out hedgehog in the early Drosophila, or you had hedgehog mutants in the early Drosophila, you got a smooth cuticle instead of one with bristles all over it. So there was these little bristly things looked like hedgehogs. So the name hedgehog, and there are two of them, both expressed in the epithelium, and they signal to the mesenchyme. So the intestine is built with a tube of epithelium surrounded by loose mesenchyme. That loose mesenchyme includes muscles and nerves and blood vessels and fibroblasts and lots of things. So the epithelium secretes material basally out to the mesenchyme. And in the mesenchyme, when the cells see that hedgehog signal at one point in time, and we think there's another trigger that we don't understand, all of a sudden it forms these little balls, these clusters of mesenchymal cells. And these clusters are what's responsible for changing the shape of the overlying epithelium. So they make first little dents in the basement membrane of the epithelium. And then later the cells on top shorten and widen. So they change shape. That automatically makes a bulge like a villus, and it's starting to grow. So these mechanisms seem to be one initiates the next, and a little cascade of events that results in villus formation. Now the clusters themselves, a different story, and all of this is by Kate Walton, who's still at the University of Michigan, a research scientist, very talented, now working with Jason Spence. But she has found how these clusters form, how they connect blood vessels, and how they're patterned. So the clusters are formed in a very nicely arrayed pattern. It starts in the proximal intestine and extends down to the end over a period of about 36 hours. And what she's shown is that patterned field of clusters, which if you look at the intestine grossly, looks like spots, well-spaced spots. That is determined by a Turing type field. This harkens back to Alan Turing, the discoverer of the Enigma code, but also did seminal work following the war, where he discovered how chemicals interact with one another. And he described this Turing field system in which cells are, it's a two-component system. The cells or group of cells secrete both an activator and inhibitor. The inhibitor travels farther than the activator, creating a zone of inhibition where another group of cells can't form. So that's patterning the clusters. And Kate showed not only that this happens, but that she began to define the signaling programs, which actually involve BMP, bone morphogenetic protein, that are responsible for this patterning. So that has been mostly our contribution in a way to the gut morphogenesis and ontogeny, if you will. The inflammation story was, if you will, a side story, which actually turned out to be very interesting, but we haven't completely followed up on it. It appears that if you reduce hedgehog in the fetus, you have a large amount of inflammation, suggesting that hedgehog proteins are really responsible for keeping inflammation low. And we were wondering whether that had implications for inflammatory bowel disease. And so we did suggest that notion, and two other laboratories took up that story and recently published papers in the adult, and we seldom work in the adult animal, mouse, that it appears that hedgehog does play a role in reducing inflammation in the colon. So
very interesting.
That turned out to be a fascinating story, which we personally did not follow up on, but turned out to be really fun to watch the story happen in the field.
The elegance and the description of some of these things, even back in the early 20th century, before we knew much about genes and other components of molecular biology, but how you really put together this stepwise dynamic of every little bit that was happening in embryos 100 years ago is just, just as a side note, I find there's so much we learn in the space that we, for some reason, forget over time, and it's nice to see. Obviously, things like
I totally agree with you. And the things that I've always encouraged my students is to read older literature. It's really very interesting. Some of the notions that people have, they see something, they don't know the mechanism, they don't know the real why, they don't know the real what, but they have a feeling that something's going on and they express that. And you can learn so much from those papers. They're very good clues to what might be going on in your system.
Absolutely. So on that note, so we just transition a teeny bit towards embryology in general. Obviously, a lot of work has happened over the last century, more than last century in sort of embryological development. Just from a 2019 perspective, where are we now? What are we, as a scientific community that's focusing on the embryo, what are the primary things we're looking for now? Is it more on the molecular side? Are we trying to reconnect, as you were pointing out, with some of these patterning dynamics that may not be, they involve genes, but of course other things, forces of mechanotransduction and membrane potentials and all sorts of biophysical dynamics. Where's the field of embryology going in general?
Yeah, that's a complicated question that actually depends on your own perspective. But I would say that, recently there's been a very large emphasis on the use of organoids. So taking human embryonic stem cells and now induced pluripotent stem cells, which are really adult cells, usually skin cells or something, that are reprogrammed to be embryonic stem cells. Once they have the capability of doing anything. So people are using these in so many different ways to model organ development and try to understand the remarkable ability of cells to self-organize themselves. They're using them for detecting certain disease phenotypes, even before they know what gene is wrong. Or even in some cases, for example, there's some examples at the University of Michigan where people have looked at cells from patients with Dravet syndrome. That's a very severe form of epilepsy. Taken human embryonic stem cells, put them in a dish, taken skin cells, put them in a dish, made them into embryonic stem cells, and then once they were pluripotent, put on other chemicals to make them be cardiomyocytes. Cardiomyocytes get together in a dish and they actually beat. And this is work of Jack Parent and others at the University of Michigan. Miriam Meisler, I think, was involved in that work. The normal cardiomyocyte cells beat quite nicely together in the dish, but the one from the Dravet patient writhe. They don't beat synchronously. So here you have a diagnosis in a way of that in a dish, and it gives you an opportunity to now imagine putting these cells in 1,000 different wells and adding a different chemical to each well and asking which of those chemicals makes the cells behave more normally. And you're into individual pharmacokinetics. So there's tremendous potential in this field for that kind of thing. For modeling organisms, people are now beginning to get organoids that model certain parts of the brain. They've certainly done it with the lung, with the stomach, with the intestine, colon. So many different organs now have been modeled, if you will, in a limited way, but
Sure.
they still have a remarkable ability to the cells talk to each other as they're making their individual structures, and they are able to self-organize, and
Mhm.
it's actually remarkable the potential that the cells have.
Yeah, I think you mentioned a fascinating thing that we think about it in terms of the cardiomyocytes, but I think there's, people are seeing it obviously in many other tissues, sort of this, what's been defined as this peripheral automaticity that, between a cell and that whole organ is the organoids, but there's these many steps as that process goes along, where, as you're saying, these cells are communicating, they're forming, they're self-organizing, and while that brain organoid might not think, there's other stuff happening as it clusters, as it creates that, a three-dimensional pattern. I'm speaking outside my domain here, but it is clearly a fascinating time on that front. And I've done some shows in the past on, sort of the bioengineering of replacement organs, and a lot of that has focused on, these pre-existing scaffolds, whether, the decellularization or bioprinting, things like that. But at the end of the day, they come from the other direction. While what you're talking about is really that, how stuff happens in nature. You start with one and you get millions or billions that form in this self-organizing, emergent fashion, which is, I think I agree with you. We're at a really unique time for some of these tools. That being said, I'm going to give you the floor now to talk about the recent paper, because it's, it's been all over the world, all over the news. Talk about what's going on there, because it, obviously extremely exciting for those that sort of know what it means. For people not familiar with the space, people are thinking, what is this? What are we creating here? But if you could take us through that paper and tell us why it's not the end of the world, and why it's really exciting.
It's definitely not the end of the world. It's so, I think we need to start from the fact that really up till very recently, and even now, we know almost nothing about what happens to the human embryo during and just after the time it implants into the uterine wall. We know that, in vitro fertilization has been around for years.
Mm-hmm.
People have consented in the past to have their embryos used in research, the ones that were going to be discarded when they were done with their reproductive needs. One has to decide whether to store the embryo, which costs the extra embryos, which cost money, or to discard them. And many choose to discard them, but some choose to donate those for research. Now, those are, let's say, normal human embryos. They've been made by in vitro fertilization. So there's a sperm and an egg union, and then a growth of the embryo. Mm-hmm. However, you can only culture these embryos by agreement across the world. There's a rule called the Warnock Rule, which was established in 1984 by a panel of lawyers and ethicists and scientists and public and religious leaders. And they agreed that one should not culture a human embryo made from sperm and egg past 14 days for the first formation of the primitive streak. Now, the primitive streak is a little ridge that you can actually see in the embryo, and it's the first sign of a process called gastrulation. Before gastrulation, all the cells are pretty much equivalent. The process of gastrulation is magical. The cells actually move, and they differentiate into one of three germ layers that is essential to build the entire body. The ectoderm, which eventually makes the skin and the brain. The mesoderm, which eventually makes all the muscles, the blood vessels, the bone, the cartilage, things like that. And the endoderm, which is going to make the gut tube and the lining of the lungs and pancreas and accessory organs. So those three are called germ layers: endoderm, ectoderm, and mesoderm. They're called germ layers. Right. They form at gastrulation. And without them, you don't have a body plan. So the agreement was that you couldn't culture embryos past 14 days. Now, at 14 days is, if you let an embryo go, is just the very beginning of gastrulation. Now, the process of gastrulation normally occurs as the embryo is actually implanting into the uterine wall. So imagine you had the embryo coming down the uterine tube, and it is basically a ball of cells.
Right.
With at one end of that ball, there's one pole, there's a little mass of cells that's called the inner cell mass, and this whole thing is called a blastocyst.
Mm-hmm.
And as it implants, that inner cell mass, which is again just unpolarized cells, becomes a cyst. It has a central lumen. It forms a central lumen. So it's like more like a balloon, if you will, with space in the middle.
Sure.
And it's just a cyst of pluripotent cells. Now, as this thing begins to implant, the cells on the side of the uterus start to flatten, and they actually turn into amnion. So that's the first formation of amnion. The cells on the other side remain pluripotent, and they will give rise to the whole embryo. After the amnion is formed, which only takes a couple days, this amnion primordium, then the cells on the other side start to gastrulate. Mm. And part of the message of the recent paper was that, and by the way, that work was done in collaboration with Jianping Fu, who's in mechanical engineering. After the amnion forms, there seems to be a cue, perhaps derived from the amnion, that tells the epiblast it's time to gastrulate.
Okay.
Now, normally, up to this point, no one had been successful in taking a fertilized egg and growing it in culture and having it get all the way to the point of forming amnion and undergoing gastrulation. And even if they had, they would have had to stop the experiment right there.
Right.
Because, again, that's the Warnock Rule, which, by the way, is not a law. It's an understanding and agreed upon rule. It's not a law in the U.S. So the slightly different tack that we took in our paper, rather than using entire embryos, we actually modeled only a piece of the embryo. Mm. So the entire embryo, as I said, was a blastocyst, and that was surrounded by trophectoderm, which is going to contribute to the placenta, and the inner cell mass is kind of held in place by a layer of extra embryonic visceral endoderm, which is also extra embryonic tissue. Those two things are missing, and without them, you don't get the placenta, the chorion, the yolk sac. Okay. You have the core of the embryo. And that core that we modeled is that initially we start from an unpolarized ball of cells. Those cells form a lumen, make a pluripotent cyst, and then we can add chemicals. What I'll tell you, there's another way to do this, but we can add chemicals to make the amnion side flatten, and then the amnion does its work to send a signal to the epiblast side to start a gastrulation. Mm. So all that can be initiated in culture with high reproducibility. And that's kind of the subject of the most recent Nature paper. But kind of taking a step back to tell you how we got there, because it's kind of an interesting story.
Sure.
This all arose from, as I mentioned, our attempts to look at how the gut lumen is actually put together, how the microvilli are established there. And so we wanted to try to use organoids, and you can make gut organoids from human embryonic stem cells or induced pluripotent stem cells. And those organoids for gut, again, are very small cysts. Mm-hmm. The inner surface is really the lumen, and that's where the microvilli will form. So we were trying to look at the very early steps in this process, and the control is human embryonic stem cells alone. The test was human embryonic stem cells exposed to stuff that make them endoderm, so gut, right? What we were surprised to see is that when you sparsely plate individual cells, either gut cells or human embryonic stem cells, they form a lumen on their first cell division. So the cell divides, and as it divides, you can see a tiny little lumen between that is shared by those two cells. So the very first cell division, they form a lumen. And that was surprising to us because the control cells did it just as well as the gut cells. I thought the gut cells would do it because, you know, the gut is basically a luminal tube. But the control cells, I didn't expect that. And that was surprising. We thought about it for a while and we said, well, okay, it does make sense because the first thing that blastocyst with its inner cell mass has to do is that the inner cell mass has to polarize and form a cyst. Why shouldn't human embryonic stem cells be good at forming a cyst? Right. And if they are so good at forming a cyst, can we find conditions where they will turn into amnion? And that's where our the first paper in the series started. Again, it was collaboration with Jian Ping. And by the way, I should mention that the lumen forming stuff was all done by Ken Taniguchi, who's now a new assistant professor at Medical College of Wisconsin. And he was doing a postdoc in the lab, and he's the one that discovered all of this. And not only this story, but discovered a very interesting story about exactly how the lumen forms between those two cells. But getting back to this particular story, Ken initially went to Jianping Fu's lab because they had been doing microprinting, and Ken wanted to make little tiny dots of extracellular matrix that would stick individual cells on a slide so he could kind of space them nicely and look at a whole bunch of them at once and also do electron microscopy on them. And so when he was over there, he met Yui Xiao, and Yui was a graduate student with Jianping, and they started discussing this whole project and discussing Ken's idea that there should be a way to make amnion. And Yui, being a mechanical engineer, said, well, you know, what if it's mechanical? And so the two of them designed 4 different kind of culture conditions that had different mechanical properties. And one of them, not the other 3, but one of them worked to make these pluripotent cysts turn into amnion on one side and blast cells on the other without adding any chemicals. So it was the mechanical stimulus, not the chemicals. The mechanical stimulus we found did cause the cells to secrete BMP, and BMP is required to make amnion. So now in the little devices that we use in the most recent paper, we flow BMP through the little device to start the amnion process. But originally, we found that the cue was probably as the embryo encounters the uterine wall, it senses a different mechanical environment than the cells on the other side that are not in contact with the wall. So what we think we modeled in culture is this mechanical force. So that was the story kind of leading up to it. In this kind of stochastic one, in the mechanical force model, basically what it is a thick gel. Okay. And you plate the little clumps of cells on that, and then you overlay that with a very thin gel. So the cells are against something that they can pull on, and then above them is something much, much softer. So only one side of the cyst, and that's the side that would turn into amnion, is under a different mechanical environment, if you will.
So elegant.
The problem with that technique was that if you had a whole plate full of cysts, only about 5% of them would remain asymmetric, meaning that the amnion was on one side and the epiblast on the other. The rest would probably roll, or something would happen. The amnion would spread around the whole cyst. So we got amnion cysts. Now that in and of itself was cool because no one's ever made amnion in culture before. So we haven't been able to study the properties of amnion. One could imagine that you could make, amnion has been used as a dressing for burns on the battlefield.
Okay.
There are some important problems with weak amnions or tears in the amnion that threaten fetal life. So you can imagine with this technique making amnion to be able to have a universal burn donor shelf. You could imagine learning exactly more about what these tears are and how to fix them. So there's a lot of reasons to be very happy that we know how to make amnion now.
Absolutely.
And one of the things Ken is actively pursuing, what are the transcriptional programs involved in doing that? But in addition, it's kind of cool to put the amnion back into the whole system, to have a system like this new one where reproducibly, you get amnion on one side and epiblast on the other, and you can study all the signaling interactions, which is what this newer device allows us to do. So it's still very much in its infancy. No one's ever made an amniotic sac before. No one's ever made amnion before. No one's ever showed a reproducible gastrulation before. So both Ken and Jianping have a lot to work on. I'm now officially retired as of July. However, I still have my hands in a few things and actively talk with Ken and with Kate Walton, who's still working on the Villis formation, and Shawang, who's at Duke working on the lengthening. So I still keep up and am busy writing papers and et cetera. So
I have a feeling you're going to keep your eye on it, and you'll you'll have a lot of input to what's going on. But clearly it looks like you've opened up the next 30 years to learning a bunch of stuff, because clearly
it's very exciting. It makes me wish I was at the beginning, not the end of my career, but there are certain advantages of this too.
Absolutely. Clearly an extremely elegant set of experiments and work leads me into the bioartography project, which clearly is a major project in its own right. Talk to us about that and what you're planning there, what you're doing, where that's all going, because I think once again it's so beautiful just to look at it, but we're in this weird world nowadays where everyone's paying attention to their Twitter feeds and whatever, but I think there's an extreme importance in sitting back and looking at the beauty of the biologic world and life, and no matter how microscopic or macroscopic these things that are happening, and your project really helps reconnect with that for the average person that might not understand all the science, but they can appreciate that red blood cell or that villi or whatever. Tell us a little bit about this project.
The project actually started in 2005. Myself and Sue O'Shea and a few other people were sitting down, Kim Lim, were sitting down to think about how to fund trainees to send them to meetings. NIH grants are great, that's what we work off of, but they're often insufficient funds to be able to send our students and postdocs to these international conferences and national conferences that are so important for them to be able to present their work, to be able to network with their peers, keep up with the latest breaking science, et cetera. So we thought, well, we see these images every day under the microscope, and they're beautiful. And what would happen if we then mat and frame some of these images and sell them to the public? Mm-hmm. And could we make enough money to send people to meetings? And the short answer is yes. We've sent nearly 100 students now over the years, a few every year, to various meetings with $500 travel awards. So it's been, I think, very successful in that regard. But the success that we didn't kind of really even think about at the beginning, but has been maybe one of the most important aspects of this, is the fact that it gives us a chance to talk to the public about the science. Mm-hmm. So here's the way it works. We get probably about 100 to 120 images. They come from all over the university. They even come from outside the university. When I go to a conference, I'll see some really pretty picture and I'll ask somebody, can you send me that and can I use it? So they all come into my office. I play with them in Photoshop a little bit, sometimes changing hues or, adding a filter and make them really pop. And then I take those 100, 120 over to the art school. And I ask 5 arts professionals in the School of Arts to tell me which are the 10 best. Those are the ones that we go with. Because as scientists, we're very attached to the scientific impact of the picture. And
Sure.
but the artists are kind of free of that. So they see the beauty, symmetry, the form, the color. So they are a better judge than we are as to what will sell. So they tell us, and we then work those up, and we have a booth at the Ann Arbor Art Fair, which draws about 1 million people over 4 days in July in Ann Arbor. And we've also sold in a couple of smaller fairs like the Tawas City Art Fair, once in Traverse City, things around Michigan. But that is the opportunity where people walk into the booth and they'll come up to a picture and it'll be pancreatic cancer. And they'll see this beautiful array of colors and shapes and look at it and be drawn in. And then you tell them what it is. And then some people will say, ew, and turn around and walk out. And others get drawn in even more. And they say, oh, really? Tell me about this. So then you have the opportunity to say, well, this is pancreatic cancer. This is the hedgehog is the signaling program involved here. And this lab is studying how the cancer cells actually initiate by aberrant hedgehog signaling or something like this. So it's your opportunity to tell them more about the basic research that's going on, kind of deflate some of those scary things, human embryonic stem cells themselves. What are they? What are induced pluripotent stem cells? How do you make them? Why do we want to mess with something like that?
Right.
What advantages? You have the opportunity to talk to them about that, evolution, the use of animals in research, all kinds of topics that are delicate. But we have the opportunity to kind of give them facts.
Right.
And we try, we do not argue with anyone. We do not try to persuade them. If they're anti-evolutionist, we do not try to persuade them any differently, but just give them facts as we have them in front of us. And, this is a really great opportunity to educate the public and to inform the next generation. Because the coolest thing is the little 10-year-old girl boy who comes into the booth and says, wow, I want to be a scientist when I grow up. Then, there's a warm feeling in the cockles of your heart. Right. So it's a really great opportunity.
I imagine that it's completely outstanding to see these reactions.
Yeah, it's so this year we're now working on a coffee table book. And we recently, with the help of Christiane Myers in the school, she's one of our jurors, she took a sabbatical and used it to put bioart on fabric. So we, for the first year, had some scarves at the booth, which were pretty awesome. Really neat. Yeah, that was fun. Very cool. So our ongoing challenge with bioart is really funding it. Sure. We do make enough at each fair to account for the supplies for the next year and to send some people to meetings every year, 4 to 6 to 8 at one point, students to meetings. But I don't take a cent for it, but I have an administrative assistant who does the heavy lifting and all the logistics that we do, including handling our website, which is www.bioartography.com. But she has to be paid. And that is the part that I have to go to the dean and to the chairs and to the, research offices of the university and try to get money to keep the program running. And that gets harder and harder every year. So that's a little bit the challenge. So we'll see how that goes.
I really, appreciate the time that you've been spending, and your time is extremely valuable. I just have one other question I want to throw at you. It's sort of our wrap-up question. It gets a little more science fiction oriented and personal, and take your time with this one. But obviously, throughout your career, you've met a wide range of very interesting people that have influenced you along this path, from Mr. Case to Francis Collins. This question, though, is about the person you would have wanted to meet if you had the chance, who's no longer here. And if you had that hypothetical time machine to go anywhere, it doesn't have to be a scientist. It can be an artist or a philosopher or wherever. For Dr. Deborah Gumucio, who would that be? And who would you want to meet, and what would you want to talk to them about?
I would have a hard time choosing between two people.
Okay, well, you have a little party. It's fine.
Okay, yeah, okay. I can have them both. Barbara McClintock. She was a geneticist who studied corn, ZMAs, and discovered transposons back in the 30s and 40s. So, way back before anyone really even knew, the structure of DNA.
Right.
This woman was so forward thinking that most of her actual theories and everything turned out to be entirely correct. But for a long time, she was, nobody believed her, to the point where she actually stopped writing papers for a while. But fortunately, that all rectified before she finally passed away. And she actually did win the Nobel Prize in 1963 for her seminal work in that area. She predicted epigenetics. The woman was brilliant. So that's one.
Absolutely.
The other one is Elizabeth Crosby. She was actually someone that probably is much less well known, but a very important figure in the history of our department at the University of Michigan. She was a neuroscientist and the only woman for a while in the neuroscience division, but she knew so much about the anatomy of the brain that neuroscientists would consult with her, neurosurgeons would consult with her before they did brain operations, because she could tell them exactly where to go in and how to navigate once in the brain. She was absolutely amazing, published widely, was active well into her 90s. Actually did meet her peripherally before I knew who she was when I put my books down on her seat in the library. I didn't know this was her seat. And somebody came to me and took my books, wrapped it away, and said, "No, Elizabeth Crosby sits here." And when I later found out her accomplishments, again, just a woman who just really pushed the boundaries of knowledge well past anybody else of her time. And she was eventually given the Presidential Medal of Freedom. Yeah.
Outstanding selections. Outstanding selections. It has been a real honor having you on the show today, really sharing your insight with everybody, as we say on the show, moving the human story forward. Thank you for everything you've done in your career. Thank you for everything I'm sure you will continue to be doing in the years ahead. It's been a real pleasure. And once again, for everybody watching and listening, it's been Professor Dr. Deborah Gumucio, Professor Emerita, Cell and Developmental Biology, University of Michigan Medical School. It's been a real pleasure having you today, and thank you so much for taking the time to share your knowledge with us.
Thank you so much, Ira. I really appreciate the chance to get to share this, and I think what you're doing is a great thing, to be able to have people have a more broad understanding of the various kinds of things that go on behind the walls of the research institutions in our country.